Well, good morning. It's really a pleasure to be included in this extraordinary meeting in a field that I am very committed. I am the medical director of the Heart Institute at the University of South Florida and have been in the field of management of heart failure for over 25 years. So my perspective on where we are today and the discussion of unmet need, I think, will become fairly apparent. I think this is appropriate to start with cardiovascular disease because it is the leading health problem in the world. It eclipsed acute infectious diseases as the leading cause of death in 2009. It causes more death, more hospitalization, and more loss of work productivity than any other disease category. I think importantly, when you look at the World Health Organization looking globally, and we're talking really not this parochial United States vision, but this is a problem that affects around the world. We don't have all the demographics from China and India and big populated areas in terms of its overall prevalence, but it's very clear that the World Health Organization has changed from acute infectious illness now to chronic diseases because it's very clear this is where we utilize the majority of our resources, this is where most of the morbidity exists, and where we have to begin to make a difference. This is just a quick capsule of the prevalence of cardiovascular disease today and the number of patients afflicted by this. In the United States, at least 1 million deaths a year, at least 1 million heart attacks a year, 500,000 strokes, millions of people suffering with chronic angina that does not seem to have therapy, 70 to 80 million people with hypertension, all these predisposing to progressive cardiovascular disease, and the now unequivocal epidemic of obesity and diabetes is going to make this problem continue to be the world's leading health problem. Right now, 1 in 4 people in the United States have cardiovascular disease. By 2025, 1 in 3. This is an age-related disease. The aging of this population is going to keep this front and center in terms of our health economy. What about the cost? This is from the Centers for Disease Control and the U.S. government and these projections, and these may be quite conservative. Right now, we spend about $280 billion in the care of patients with cardiovascular disease. That sounds pretty impressive, but the most impressive data are the projections just based on the aging demographics in this country, that the age-associated use of resources is going to triple that number by 2030. It's a prohibitive cost. It's an absolute mandate to get aggressive and find new things. This trend of cardiovascular disease being the leading cause of death in the United States for the last 100 years is fairly assured to be continued for the next several decades based on this prevalence and our current inadequate therapies. I'm going to talk to you specifically about my area of interest because I think it's going to dominate the cardiovascular field, and that's heart failure. The first line, I think, should grab everybody's attention. When you look at CMS data, there are more days spent in the hospital for the care of patients with heart failure than any other diagnosis in any other field. This is large hospital, small hospital. This is demographics from around the country, and I think this is really why this area has to be a target for regenerative medicine. Right now, we think there are 7 million in the United States afflicted with heart failure, at least 20 million in Europe. And again, we don't have the numbers of this big explosion in China and India, Brazil, and big populated areas. Half of them have this reduced pumping function. So as you work down the scale, we're talking millions of people as a target audience for new therapies for heart failure. I think that we're seeing more cardiovascular disease in those populated areas, largely from atherosclerosis, but Chagas disease in South America. This is an endemic problem around the world. This is a breakdown of how we get to how many people have advanced heart failure, and think about the concept that let's don't treat this as usual. They want you to work with the sickest patients and then prove that it works and work backwards. But this as a preventive strategy is a tremendously appealing concept, but I think as you look to the bottom of the slide, you can see that we're easily, right now, very close to 250 to 300,000 patients in the United States with this. Here's why this is so important. On the left-hand side of your slide, we don't have any therapies. When people get to an advanced stage of heart failure, we have almost nothing to offer. Imagine 20% of the people who come in to get improved and go home will be back within a month, and half of them will be back within 6 months. We absolutely have nothing that changes the natural history. Once a patient reaches that phase, and I assure you, 550 new cases a year and people that are just becoming more refractory, we've kept them alive with defibrillators and other strategies, but the number that are getting into this advanced category is going up dramatically. And look at the mortality on the right-hand side. A third of people admitted for heart failure for a hospitalization will die within a year. This is profound impact and a disease that I think is very targetable. So what do we have? I would submit to you that we have an inadequate pharmacologic treatment paradigm. The #1 drug recommended around the world are ACE inhibitors, and the absolute reduction in mortality is 2% per year. That's our best drug. That's why these people are progressing, and we're seeing this go on. This is referred to as the graveyard in the pharmaceutical industry. 13 drugs since the year 2002 have gone to phase 3A trials without being successful. We are using survival, we're using composite endpoints, but I think we've based all of our therapy on biomarkers, and that is our targetable strategy, but we have failed in the last 10 years to have a new drug and a new game changer. I think that experimental drugs, I don't see much on the horizon, but I'm not completely nihilistic. But heart transplantation as alternative is, as people would say, epidemiologically trivial. 2,200 in the United States and 250,000 in need. So that's not going to go anywhere. Mechanical cyst devices, I think, are still cost prohibitive because we have to think globally. This is not a U.S. market problem. I mean, the numbers from an industry and an investment point of view, obviously, to get outside the U.S. is where you're going to see an explosion in terms of the millions that can be treated. So I think biologics have one other huge attribute, especially over pharmacologic treatment, and that is that compliance with the drug regimen is difficult. In many surveys, when you get down to the people taking the right drug at the right dose and being compliant, you're looking at about 20%, and you wonder why we're not getting there. These biologics have the opportunity as a single administration. It's not based on compliance. You don't have 20% dropout because of drug adverse effects. And so I think this can change the recalcitrant group of patients that keep coming back and forth. And the concept I talked about of readmission is probably going to become one of the central. You're probably aware that Medicare now has taken that as the #1 target because of this frequency of readmission and the costs associated as the target that they will cut reimbursement if you don't improve that strategy. Well, we don't have much on the horizon to say we can change that right now. And so I think for all these reasons, I think heart failure is without a doubt the important strategy in the cardiovascular market, and the cardiovascular disease, the overwhelming #1 health problem in the world. Thank you.
And Les will play an active role in our panel, making sure that, medically we're accurate as we go to him. The first speaker today is going to be Mesoblast. So I'd like to have Professor Silviu Itescu come up and talk a little bit and answer the questions in terms of mechanism of action, the clinical program, and the SWOT. How does Mesoblast score?
Thank you very much. So I'm going to try to be brief. I've been asked to be very brief. For us, we're developing a platform technology based on mesenchymal precursor cells, the earliest precursor of the mesenchymal lineage. And it's very important for us to think about mechanisms of action if we're going to be developing products along different lines and different indications. As diverse, for example, as immune-based therapies versus cardiovascular therapies versus orthopedic therapies. So our strategic product development is guided by mechanistic studies preclinically. The concept is that the cells that we're developing respond to signals from areas of inflammation and or tissue damage, so that the type of response of the cells and their secretion of paracrine factors depends on the signals that they receive within the specific tissue that they're encountering. So, for example, in damaged cardiovascular tissue and various other tissues that are ischemic, interaction with activation signals result in release of factors that are responsible for stimulating blood vessel growth and maturation and reversal of endothelial dysfunction. Other factors, and we can define these specifically, other factors enhance survival and improve function of cardiac muscle cells, cells of other tissues including nervous system, bone-forming cells, and cartilage-producing cells. The concept is enhanced survival, anti-apoptosis of the endogenous cell types. More interesting data that seem to support the use of these cells systemically for diseases of inflammation include the induction of polarization and conversion of a pro-inflammatory monocyte phenotype to an anti-inflammatory monocyte phenotype, and in parallel, inhibition of activated T cells, particularly the autoimmune-inducing Th17 cells, to an anti-immunologic induction of Tregs. And so you can start to see how the response and the effect on these specific targets might give you the ability to develop products that are specific to very, very precise disease states. Now, in order to take advantage of these particular cells that we're working with, mesenchymal precursors, we understood there were two technical advantages. Number one, that the cells are very easy to expand in large numbers in vitro from a very small number of starting material. And to do that, we start with a very pure, homogeneous, immunoselected population of cells based on understanding their surface markers. And secondly, by expanding these cells, we end up with effectively an industrialized manufacturing process. And we do that because we take advantage of a second property, and that is that they seem to be relatively non-immunogenic. The reason why they're non-immunogenic is also well understood scientifically. They lack a whole range of surface molecules that typically are necessary for induction of immune activation, and we all know that transplant rejection is driven by these molecules which are missing on these cells. And so they induce almost a tolerogenic effect on T cell activation. So if you take those two concepts, easy to expand and relatively non-immunogenic, you can see how you can industrialize a large-scale manufacturing process. The starting point, I say again, is to understand the surface phenotype of the cells very precisely, the exact markers on the surface of these cells in vivo, and to be able to extract the cells out using monoclonal antibodies to a very pure homogeneous phenotype. And here we use an antibody called STRO3 to define these cells quite precisely, then expand them in a GMP facility, and we end up via cell banks, master cell banks, and then batched expansions with really a process that looks for all the world like pharmaceutical drug development. The regulators are comfortable, our partners are comfortable, and more importantly, we're comfortable that the final product has very precise release criteria with batch-to-batch consistency. And that means that we can minimize the cost of goods on the one hand. We can optimize consistency from product to product and patient to patient, which obviously has a great impact on outcomes in clinical trials. And it gives great comfort to the regulators to know that what they're regulating when finally you complete a phase 3 program is a well-characterized, well-understood, consistent product from a safety perspective. So in terms of our cardiovascular program, you've heard from Les the great medical need, and I come from a cardiac transplant background myself, so I certainly understood the limitations of heart transplants in this industry. And so the concept from our point of view is to have a relatively easy-to-use, very potent, and commercially viable product that is cheap enough to make, that is appropriately reimbursable, and can be used in a large market segment like congestive heart failure. On the basis of substantial preclinical data, and I won't show you the data here, but at least 6 large animal studies and probably another half a dozen small animal preclinical studies which gave us comfort around proof of concept, around mechanisms of action, and around efficacy and safety and dose ranging, we progressed to a phase 2 trial in congestive heart failure, which was a randomized 3-to-1 study in 60 patients with quite advanced heart failure ejection fractions of 35 and below. And the reason we engaged in this patient population is because that's the population where, despite optimal medical therapy, there's a relatively high event rate that can be seen over a 12 to 24-month period, and where there's very clear mandate from the FDA about what they expect to see in any product, not just a biologic, but drugs, devices, any product, if you're trying to have a product approved for this population. What the FDA absolutely expects you to demonstrate is an effect on mortality and heart failure hospitalization in one form or another, whether it's some type of composite endpoint. And this is the population where you're going to see, if you're going to see an effect, this is where you're going to be able to see an effect in a relatively small number of patients. Although, to be fair, the primary objective of this first study that we did was primarily safety, and we were hoping to see some effects on surrogate endpoints. The cells were injected by a single endomyocardial injection with a Johnson & Johnson Nogastar catheter, which has been extensively used in a variety of clinical trials. As a primary endpoint of safety, we had no problems. We tested 3 doses, low, medium, and high, based on dose-ranging studies that we'd seen in sheep. And what we were particularly quite astounded to see, that in this tiny little study with very small numbers, we ended up now 3 years of follow-up with a very significant effect on the primary endpoint of a composite of heart failure, hospitalization, and mortality. And what you see in the control population over a 3-year follow-up is about what you see in the general published literature in control groups of these types, where the time to a first event is, by 36 months, by Kaplan-Meier, approximates about 30 to 40%. And in a 12-month period, you see about 20% of patients progressing to a hospitalization event or death. And you still see that at the highest dose, a single injection of our cells has protected the patients from having any event over 3 years of follow-up. These are really astounding data. If we were to see this in a phase 3 trial, we'd have an approved product. Now, the 2 intermediate doses gave results that were somewhere in between and were clearly not as significant as the highest dose. And let me show you why I think that is. So in order to try to understand mechanistically what's going on, you look at surrogate markers. And we look at end-systolic volume and end-diastolic volume as the obvious 2 surrogate markers, which really show you whether or not you have an effect on the volumes of the myocardium. What you're really trying to do is reduce the volumes in both systole and diastole if you're going to remodel the heart in a positive way. And you look at 6 and 12 months, and what you see in both systolic volume and diastolic volume is clearly a dose effect, where the lowest dose had no effect, the mid dose had a small effect, and the highest dose had the most pronounced, significant effect at both 6 and 12 months. And if you believe that these volume effects are real, and you believe that that they're reflecting a remodeling process, seeing them at 6 and 12 months should be predictive of what you see over the subsequent 18 to 24 to 36 months in terms of survival. And we think that these effects on remodeling were the explanation and predictive of the subsequent lack of hospitalization events out to 36 months. So the conclusion that we have at this point is that the cells are safe in both preclinical and clinical studies. In fact, we haven't seen a maximal tolerated dose. We've seen no cell-related adverse events. A single administration at a particular dose, so far the dose that we've identified is this 150 million cell dose, sustained improvement in hospitalization and death over a 36-month time frame, and results in concordant improvement in various surrogates in comparison to controls. So we're at the moment in a partnership with Teva Pharmaceuticals, and at the moment advancing. We're proceeding with a 1,700 patient phase 3 trial in this group of patients, expected to commence by mid-year. I think that the rationale for this 1,700 patient trial, we might talk later, is a 1-to-1 randomization. It's powered for an effect on survival and hospitalization that anticipates a much weaker effect than we've seen in the phase 2. But the primary driver of numbers here is not just the powering, but actually the expectation by the regulators that there should be roughly a minimum of 800 patients exposed to the drug prior to regulatory approval. And that's really the rationale for the way this study is set up.
Perfect.
Thank you.
So you're certainly going to want to come and listen to Mesoblast this afternoon and hear a little bit about some of the other clinical programs, particularly in orthopedics, rheumatoid arthritis, macular degeneration. So this is kind of a multi-product company with multiple clinical trials. Rhonda Bartel is here. as the Chief Scientific Officer from Aastrom. Aastrom recently changed gears. They made a very tough decision, which was not to go forward in critical limb ischemia, but to move ahead in dilated cardiac myopathy. So I'm looking forward to hearing about the same questions, and also tell us a little bit about DCM and why DCM. Thank you.
Oops, wrong way. Okay. First, I'll talk about a little bit about what the product is that we're looking at in the cardiac space, which is Mile SLT. It's a unique multicellular therapy, and it is a patient-specific or autologous therapy. As you can see across the top line is the composition of bone marrow. We start with a small bone marrow aspirate from the patient, about 50 cc's. We then go through a process in which we debulk or remove the red cells, and then we put the mononuclear cell fraction in culture for 12 days. And what happens during that 12 days is you see a reduction, not an elimination, but a reduction in the lymphocyte and other hematopoietic cells like granulocytes, for example, about a 5-fold reduction. But what you see in the other components, in the monocyte and the stromal cell fraction, is in the CD90 positive mesenchymal stromal cells, we see about a 50-fold increase in expansion cells that are very similar to what you just heard about from Mesoblast. And then in the monocyte fraction, we have both expansion of the monocytes as well as differentiation into, on this side it's called the CD14 macrophage, which are actually M2-like or anti-inflammatory macrophages. So at the end, the product that we deliver to the patient is a multifaceted approach. So we're looking at many ways to hit on the target. In terms of the mechanism of action, backwards for some reason. So if we look at the two effector cell types, which in the bone marrow, these cells are less than 2% of the total population. And in our final product, they constitute between 40 to 50% of the cells at the end of the day. If you look at the activities, both of these cell types secrete very similar soluble factors. They have anti-inflammatory cytokines, predominantly interleukin-10 and interleukin-1 alpha receptor antagonist. They also secrete a variety of matrix metalloproteinases and the inhibitors of those enzymes, as well as angiogenic factors, VEGF, adiponectin, basic FGF, as well as numerous others. But where we really see the differentiation is what I call the cell-based or cell-dependent activities. These are things, if you think about the soluble factors, these are things you might be able to approach in a cocktail in a pharmaceutical method. But the cells need to deliver these at the right place at the right time. And the cells also convey activities themselves, that you need the cell there itself to actually see the effect. And when you look at what are the cell-dependent activities of the stromal cells, they fundamentally make and maintain extracellular matrix in terms of the cell itself instead of secretory factors. On the side of the M2-like macrophages, they actually do a lot in terms of cleaning up tissue, removing inflammation. And one of the more recent properties we've actually discovered with these cells is they very actively take up oxidized LDL and actually efflux cholesterol. We've shown data in vivo that we can load the cells with oxidized LDL and actually see secretion of cholesterol. So if you take all of these activities combined and say, what really are the panel of biological activities that this product brings to the patient? It's remodeling of extracellular matrix, it's promotion of angiogenesis, it's protection of endothelium, and modulation of inflammation. I always like to use the analogy if you're trying to take the hunter analogy, unfortunately, but if you're trying to address a problem, you can either take a rifle approach of a single cell type, very specific, very well characterized, or in the terms of a patient-specific product, can you come at it from a multiple angle and try and address? These are bone marrow-derived mononuclear cells, which are the reservoir of the regenerative capacity, and we're basically setting them into a wound environment in culture for 12 days. So the fact that you get a regenerative population of this is not really too surprising. So how do you take this to the clinic, and where are we with that? So we are currently in a phase 2B trial, which is called EXCEL-DCM, which is in ischemic dilative cardiomyopathy. The objective is to demonstrate the safety and the efficacy. We've done 2 small studies, 2 phase 2 studies. One was a surgical approach. The other was very similar to this study I'm going to talk about here. It's a randomized 1-to-1 placebo-controlled study. Again, using the Noga catheter for injections. It's a series of 20 injections in the border zone or in the electrically active but non-contractile part of the heart. We're looking at 30 sites across the U.S. The investigators are a mix of interventional cardiologists and heart failure specialists. In terms of the patients, the age range is 30 to 85. They must have ischemic dilative cardiomyopathy with limited options for revascularization, ejection fraction less than 30, and New York Heart Association Class 3 or Class 4. The trial is a total of 108 patients. The primary endpoint, very similar to what you've already heard, is we're looking at all-cause death and hospitalization admissions and ER visits for IV. So basically preventing hospitalizations and heart failure. The secondary endpoints, there's a collection of clinical, functional, and biomarkers. The assumptions that we made going into this are the treatment effect that we see in this phase 2B would support a phase 3 of about 300 patients. One thing I didn't mention is we do have orphan drug status for this indication. Sorry, I lost my place. We're looking at this to take about 1 year to recruit and 1 year to enroll. And that this 300 patients will be a sufficient safety base with the other experiences we've had in clinical trials. Okay, so why do we think this is the thing to do? We did a market research study, and this study included a mixture of interventional cardiologists and heart failure specialists. And the results from the indicate that we are really looking at the right people. You know, these are the right physicians to talk to, and this is the right approach into the clinical space. The product actually meets a well-defined clinical need. You have on one hand people who have pharmaceutical therapy, revascularizations, device therapies, and then there's a gap between there and where you get to either an LVAD placement or heart transplant. There's a well-recognized space in the middle there where this sort of therapy could be appropriate, and this is why we're pursuing it. So looking at the numbers on the bottom you see are actually post-launch numbers in terms of where we see the market with DCM. It's about 120 to 140 million patients. I'm sorry, 1,000 patients at this particular time. So we look at this as a very exciting opportunity to look at this with Mile SLT and the multicellular therapy.
Terrific. Thank you.
Sure.
So, just think for a second about Aastrom versus Mesoblast. Mesoblast is using a homogeneous pills in a bottle approach, off-the-shelf ready. Aastrom, company's core expertise is clearly manufacturing using an optimized, heterogeneous, autologous population. Orphan indication, much smaller trial, really an interesting strategy and very, very different than what Mesoblast is doing. And I hope we'll have time at the end to talk about this. One of my favorite companies, I want to ask Marc Hedrick to come up and talk about Cytori, which is really an interesting company because while it's autologous using your own cells, it's on-site processing and it's got an allogeneic-like cost of goods model, so very much in between the line of allo and auto. Thanks for coming, Marc.
Good morning, everyone. Jason, thank you for inviting us. Happy to be here, and I will be brief. So what differentiates Cytori from some other types of cell therapies? I think one of the principal differentiators is that while the product that we deliver to the patient and the doctor is a cell therapy, the product we take through the regulatory agencies is a device. And that fundamentally changes who we are as a company and the things we have to do from a regulatory and clinical perspective. And that really differentiates us, I think, from many other companies. that cell processing goes on at the bedside. It can go on in a centralized facility in the hospital or right there in the cath lab or in the operating room. So what does that mean about our technology in terms of the cell therapy? It's autologous. It's a mixed population of cells that are not cultured, so it hasn't gone through a process in a laboratory of being cultivated. Because it's a mixed population of cells, that you provide not only a variety of different paracrine factors, but a diverse group of cells to deliver those paracrine factors, and I'll show you a little bit about that in a moment. The process is patented. We have about 50-plus patents around not only the process, but the devices and the uses of those cells and the composition of matter. Now, in the interest of Jason's point about a SWOT analysis, so what's a challenge that we have being kind of real-time autologous? One is making it easy for the doctor. And so we've treated about 5,000 patients around the world. We've treated about 100 patients with intravascular delivery, many of them cardiologists who deliver that. And we're confident, in fact, we're doing this now, that we can make it completely seamless for the cardiologist. The cardiologist, if they want, can see the patient in the clinic, schedule the patient for a cath and a cell harvest, and show up in the cath lab, have a syringe of cells that they can deliver to the patient. And that's the goal with respect to our cardiovascular therapy. So just a couple of quick key points about the mechanisms of action and the types of cells that we obtain from adipose tissue. So these cells originally come from adipose tissue. When we eat a lot, our fat compartment expands. Where does that expansion come from? It basically comes from this population of cells that you see on your left. And it's a very diverse, unique population of cells. And each cell seems to do something particularly well. For example, in reversing the effects of radiation injury, the endothelial cells appear to be relatively important. To differentiate these cells into nucleus pulposus cells in the spinal disc, in which we've done in 2 large animal models, the stem cells and the CD34 positive cells seem to be the most predominant cells. And then for treating ischemia reperfusion, the tissue macrophages may play a particularly important role. So diverse cells elaborating a variety of paracrine factors, and the other 2 speakers did a terrific job of summarizing some of the common themes with respect to cell therapy in terms of paracrine delivery, anti-inflammatory properties, differentiation. I think those seem to be conserved in almost any type of cell therapy as common mechanisms of action. Initially, 10 years ago, when we started working in this area of stem cells from adipose tissue, the original idea was, let's figure out what the right cell is, and let's grow it up in a petri dish, because let's really concentrate it and try to use it. And what we found is that those cells, almost inevitably, that were cultured over many times, didn't work as well as the mixed population. So we kept going back to the mixed population. We just felt like, on the whole, that had better efficacy while maintaining a strong safety profile. So what are we doing clinically with respect to cardiovascular disease? So this really summarizes where we are overall in our program. One of the key things, and I echo what the other speakers have said and what Jason said, the key is having great data, and we started off with trying to develop robust clinical trials. Both of our initial pilot trials were double-blind randomized, prospective trials, small numbers of patients, one for heart attack and one for patients that have heart failure related to chronic myocardial ischemia. Basically, we had a lot of preclinical data, but we weren't really sure which clinical would actually show the best result, the best safety profile. So we made 2 small bets, and it showed that cell therapy and cardiovascular disease for AMI and for chronic myoischemia were both safe and both showed strong signals of efficacy. So based on those 2 preliminary studies, we're now in a U.S. phase 2 called Athena, 45 patients at 6 sites to investigate the use of this mixed population of cells from adipose tissue for patients that have heart failure and reversible ischemia by SPECT. And we're well into enrolling in that study, and we're actively discussing with FDA what a pivotal trial might look like to follow on that phase 2. We took a little bit different approach in Europe, in that we're actually in an over 200 patient, up to 216 patient pivotal study for heart attack in Europe, and we're actively enrolling in that. And then finally, just briefly, let me touch on economics. How would this work economically? We've been in the market now selling products for about 6 years. There's a piece of capital equipment. We're continuing to bring that price down, and we think ultimately we'll bring that price well below $100,000 for the cost of goods, perhaps around $10,000. Gives us a lot of flexibility to get that technology and penetrate hospitals and clinics around the world. The key value driver is the consumable, and without any scale whatsoever, and roughly a $10,000 per consumable, we can be at gross margins that are around 90%, and we have a lot of room to grow with scale. So that's the overall economic model. So with that, thank you very much for your attention. Jason, turn it back over to you.
So one of the points that Marc didn't make, and I want to discuss is, and it took me a long time to figure this out about Cytori, is this a device or is this a biologic? And it turns out that the current trial is going through the PMA pathway. So the phase 2 U.S. trial is actually called a pilot study. So my understanding is that that sets up Cytori to do one modestly powered phase 3 trial, provided that they have an acceptable endpoint with the FDA and a p-value and be approved versus a traditional BLA approach that would require 2 trials. So it's really a kind of a unique attribute of Cytori that really positions them, depending on data, to be right neck and neck with Mesoblast as one of the first approved therapies. So I'd like to ask Dr. Rhonda Marban to come up. I met Linda about a month ago out at Cedars-Sinai. Linda and her husband, Eduardo Marban, who's a very famous cardiologist, are really pioneering something very different. And when I got involved in Capricor, I was really struck by this company. I was struck because Linda's one of the only people I've ever met who's made a transition from autologous to allogeneic. She also has done a lot of experimental work, or Capricor has done experimental work about cell delivery and talking about, you know, do you have to deliver cells via NOGA? Could it be IRA? Could it even be systemically? So this is a really interesting company. And then I guess the third element that just really impressed me about Capricor is what's the best way to fix the heart? You know, how about using heart cells to fix the heart? Is there a language among the cells in terms of their cytokine expression profile that makes sense? And so I think when we look at a second or third generation of cell therapy, because for me it's no longer a question of if, it's just when, and we know when because we can calculate the timing of these trials. But when we think beyond the first generation therapies, Capricor looks ideally positioned to be a second or third generation therapy. Thanks, Linda.
Thank you very much. So good morning. Thank you very much for having me here today. You know, I think I'd like to, wrong way. I'd like to speak directly to Dr. Jonas, who spoke this morning about how we have to build a commercially viable product along with a clinically applicable product, one that actually will heal the heart and send patients back to their daily lives. Shown here, I show the approach that Capricor has taken and why our CDCs, cardiosphere-derived cells, are unique in cardiac cell therapy. If you actually look at what we're doing, we're taking a cell that is now allogeneic. Our proof of concept work was published recently in The Lancet that showed that autologous CDCs reduce infarct size measurably and generate new myocardium, suggesting that we actually are repairing the heart. However, we found that the autologous cell model was clunky, both from a manufacturing but also from a cell delivery perspective. And so we moved into an allogeneic cell. Now this product is off the shelf. It's frozen. You take it from the bag, and within 90 minutes, the interventional cardiologist can infuse it down a coronary artery. So again, this is another differentiator of Capricor. While the intramyocardial delivery systems are appealing in a certain way, you think you're delivering the cells right to the muscle. In fact, it's a more natural process to deliver the cells down the infarct-related artery. We now have a new program, which I'm sharing with you for the very first time this morning, in which we're going to be doing triple vessel infusion. That means each of the coronary arteries will be infused with a dose of cells such that we'll have coverage of the entire heart with our cell product. Very exciting recent approval from the FDA. Look for the dynamic trial to be recruiting soon. And finally, our cells are derived from the heart. So our original concept is, since it's a progenitor cell, and I think it's very important to differentiate between a stem cell and a progenitor cells, our cells actually speak the language of the heart. They're destined to become heart cells, not just myocytes, but blood cells and interstitial cells, such that the heart recognizes them. However, that's not the important mechanism, as I'll talk to you about in a moment. So I'm a scientist, and so I always have to have my busy word slide that says, the fancy markers that the cells express and do not express. But what this cell tells you is that our cells are very different from the other cells that are out there. Our primary markers are that they're CD105 positive. That's a TGF-beta receptor. We feel that that's a very important signaling molecule in terms of the mechanism of action of the cells, and they're of intrinsic cardiac origin. We don't have very much CD90 in the cells. In fact, we have a recently published paper that shows that there's an inverse relationship between the amount of CD90, the primary marker in most mesenchymal stem cells, and the effectiveness of our particular cell population. So less is more in this situation. And they're not myofibroblasts or fibroblasts-like because we don't express much of these molecules called DDR2 and SMA. Finally, they're not cardiomyocytes either. They don't express any of the markers that we all know are recognized in cardiomyocytes, intracellular proteins. And what the most important part of our cells are is not only what they express, but what they do. And what they do is that they shrink scar size and increase viable myocardium, that is contracting tissue of the heart, which has been shown both in clinical studies through the Caduceus study and also in many, many published preclinical studies shown below. So if you need some light nighttime reading, make some notes of what I've quoted below as our studies. Finally, our cells are multipotent and clonogenic, but they don't have to engraft long term for the benefit. And the cells are small, so we can put them down a coronary artery safely. What I'd like to say after all of these large words is that our CDCs function as a local drug delivery system. Why do I say that? Because we know that our cells disappear. 4 weeks after they're delivered, they're gone from the heart, but the effects remain, and we have clinical data now out beyond 1 year that show that the scar size has decreased and that there's an increase in viable myocardium. We think the cells are inducing what we like to call a role model effect. It's telling the cardiomyocytes that are there to stay alive, reducing apoptosis, which is programmed cell death. And then they're also recruiting endogenous stem cells to the site to make things better. And they're not bone marrow cells that are being recruited to the site, but actually stem cells that are held within the niche of the heart itself. So it's a very exciting mechanism. Our clinical trial program is active. We have 2 trials that are active. One is enrolling right now. It's the All-Star trial. It's our first trial. It's a phase 1/2. The FDA asked us to go back after Caduceus, which I've told you a little bit about, and prove that the allogeneic cells were safe to give to patients. And thanks to Silviu and his group for paving the way for us in terms of showing that an allogeneic cell does not induce an immune response, our barrier to entry was pretty light, and we've been able now to demonstrate that our cells, like theirs, are not immunogenic, and we have patient data to confirm that. This trial is enrolling at this time. We also just got, and I alluded to this a moment ago, a trial to begin in advanced heart failure patients. So we're looking at patients with dilated cardiomyopathy, such that Rhonda just talked to you about, but also ischemic cardiomyopathy. And we're looking at those 2 groups separately within the same trial to see if there's a difference in how the cells treat those 2 different but same endpoint disorders. You recall from Dr. Miller, heart failure is heart failure once you get to that advanced stage. And so this trial, look for it to begin recruiting soon. It's going to be a phase 1 pilot study. Finally, I'd like to talk to you about our regulatory strategy and how we're going to move this finally into patients. And our phase 3 plan, of course, as we all know, I think we realize that no matter what beautiful surrogate data we have, whether it's changes in volumes, changes in infarct size, or changes in ejection fraction, we're ultimately going to have to show that this therapy extends life, reduces hospitalizations, prevents MACE, and all of those other things that we want to do so that patients can go back to work and go back to their families and live longer. And that trial will likely be a large 1,800 patient worldwide trial beginning in 2016 with a MACE endpoint. We expect to do that trial with a pharma partner who we're in conversations with at this time.
Terrific. Thank you, Linda.
Finally, just one more point. I just have one more slide. You're not done with me yet. When I sit back and I look at Capricor and why it's different from our competitors in the field, we have a unique mechanism of action by the reduction of infarct size and the generation of new myocardium. We have an ease of use. It's an off-the-shelf allogeneic product. We use donor hearts that come from OPOs, organ procurement organizations, such that they're transplant ready and have gone through all of the qualifications, so they're safe. We have a scalable manufacturing model that we can make thousands of doses. Our projected cost of goods sold will be under $1,000 by the time we reach commercialization, such that we have a really large profit margin. We have a very broad IP portfolio, separates us from the other players in this field, and we have a proven scientific and management team that are both here and at home working hard in Los Angeles. Thank you so much for your time this morning, and I'm available for questions today.
So let's start to ask a few questions. We have a few minutes remaining, and I guess I'd like to first start with raising capital. And Silviu, one of the things that separates Mesoblast from just about every cell therapy company in the space is the company's origins and roots, your ability to raise capital, your partnership with Teva. So I'd like you to talk a little bit about just what your vision was for raising capital, how you got the company founded, how you convinced Cephalon to really buy into the program. And now with the transition to Teva's management, clearly Jeremy Levin has said that he's behind Mesoblast. But we know that there are some interim looks. Tell us how this 1,700 patient trial may kind of roll out and what that means financially. Teva's, I believe, picking up the cost of that.
You've asked a lot of questions.
Sorry.
Complex questions. So a little bit about, I guess, our financing strategy. So it was clear to us at the outset, I founded Mesoblast in 2003, that sort of time frame. It was clear that if we have aspirations of using cell therapy for a number of areas, cardiovascular being one, but obviously a very expensive area, you have to be really well funded, and you have to learn from the experience of successful biotech companies on NASDAQ that have subsequently gone through and are now selling products. And whether it's Genentech as the leader in the antibody space, or whether it's Regeneron today as one of the standouts in biotech. And the 3 or 4 major areas that we focused on was a platform technology that gives us multiple products, so that we're not reliant on success or failure based on one outcome. Secondly, sufficient cash to allow us to unlock the value of our IP, so that if you assume you've got good IP, you need to have enough cash to be able to unlock the value in a parallel time frame. Otherwise, no matter how good your technology is, you just never know how well it works. And thirdly, I think we took the view that you can't do it all by yourself, both at the level of manufacturing and at the level of distribution. So we worked hard to bring partners into the equation at both ends. And on the distribution side, Cephalon was the first partner we entered into an arrangement with. And Lonza, we have a very special relationship at the manufacturing level. So all of those things are irrespective of the kind of technology we have, but it really underpins our ability to drive our programs. In terms of cash, I guess by taking the view that we needed to have enough independence along the way to execute, we've raised capital several times. And most recently, our last financing was done about a month ago. We did raise about $170 million from very large, substantial global institutions who are well versed in the biotech space and are household names here in the States, and they support all the large biotech companies that are moving through in pre-revenues through to new revenues. And I think it's really what I'm saying is that we're a company that's being built on the foundations of traditional biotech development. It's a new industry, but we need to learn from the rest of the industry. In terms of your question about Teva, Teva are a great partner. Teva is in a transformational mode, I guess, from being the world leader in generics to being far more involved in branded products. Recently in December, they've stated what their core areas are, and of course cardiovascular medicine is not one of their core areas, but they looked very hard at the Mesoblast technology and have made some pretty clear statements and decisions around the strength of our technology and the strength of our partnership. And data speak. So They're committed to this phase 3 program. They're funding it. And I guess the interim analyses along the way, as you would expect when you go from a 60-patient phase 2 trial to a 1,700-patient phase 3 trial, is not unusual. I think any judicious pharma company would like to see a variety of interim data points in a trial that's otherwise an adaptive design. I think that's appropriate.
So one of the things that we've talked about is that Teva is obligated to fund phase 3. They're not obligated to fund phase 2. Yet my understanding is in Australia they're actually funding a phase 2 trial that's a STEMI-induced CHF. So can you help us understand why Teva decided to opt into that trial and what they asked you to do about the trial design?
Yeah, so our other major product that's being partnered is intracoronary post-large MI to prevent heart failure. Slightly different strategy at the earlier end, as much like the other folks on the panel are talking about. We were doing a relatively smallish phase 2A trial of about 90 patients, and Teva said it's not adequately powered to give the right kind of signal to be able to then make decisions on phase 3 outcomes. So they insisted on enlarging the trial to about 225 patients and contributed funds to that enlargement beyond their contractual obligations. But I think, again, it attests to the good partnership between a smaller company and a larger company where if you're aligned, your objectives should be the same, to accelerate and fast-track programs in order to see signals along the way as quickly as possible. So you can make adjustments to your program to commit more funds potentially. You have to resize, etc. Those are the kind of decisions that pharmaceutical industry players make all the time with small molecule drugs. So what we're seeing here is the same logic being applied to a whole new industry.
And my last question for you, Silviu, and we'll kind of get into this with the other panelists, is that, there's clearly an argument here brewing between what happens with a homogeneous cell, say in your case a precursor MSC, earlier in the lineage but very, very trophically potent, versus the heterogeneous population mix. So can you help us understand why you decided to go the homogeneous route versus heterogeneous?
We've taken a view from the very beginning that the greater the understanding of your product, the better the characterization of your product, the easier it's going to be to identify signals, outcomes, and have easy discussions with the regulators. And that's kind of the standard pharmaceutical approach to defining the active ingredient in a heterogeneous mix, let's say, of natural compounds, identify the active ingredient, concentrate it, and deliver that. I guess with cell therapy, there's no doubt that living cells have an effect on their target tissues, and that's what we're all talking about here. The question is whether that living cell that you're delivering as a product needs to be combined with any other living cell, or whether it does its best effect when it encounters the living cell that's damaged in the tissue of need. And our view has been that's the optimal approach. If we can optimize the cell type that we're injecting, and first generation is really a simple, optimized, expanded product, but a second generation product might very well be genetically modified or modified cell surface to allow us to target specific tissues. And so if we understand our cell optimally, then we can modify it for life cycle extension quite easily.
So let me turn that question to both Rhonda and to Marc. And Marc, let me ask you to address. Cytori decided to go through the adipose route. And clearly, could you talk a little bit about what the number and the amount of stem cells present in that tissue is versus the marrow? And then can you address a little bit why the heterogeneous approach? Did Cytori ever consider trying to pull or optimize the population in the way that Aastrom is?
So the answer to the first question is regarding cell number, adipose versus bone marrow. If you look just at the stem cell population, the multipotent clonally derived stem cell, where you take one cell, grow it up in a petri dish, and it can differentiate into multiple cell types, that's about 1 to 5%, depending on the donor, from adipose tissue, from the extract that we get from our system. If you look at a similar population from fresh bone marrow. Depending on the age of the donor, which is critical, you're somewhere on the order of 1 to 50,000 up to 1 and 2 million based on published results. So we're kind of in the neighborhood of 100 to 1,000 times the number of these pluripotent cells per volume of tissue. So from a concentration perspective, for whatever reason, they seem to be higher in adipose. Now, why did we pursue a path towards the mixed population versus a cultured or more clonally derived population? Really goes back to about 10 years ago when I was writing my first R01 grant at UCLA, and our original idea was to culture the cells to improve their ability to make bone in patients that had craniofacial defects. And so we cultured the cells over and over, tried to get those cells to form bone, but we just really couldn't find good bone formation in our preclinical models. Then we looked at the mixed population of cells and found they worked quite well. And so we looked at cartilage and muscle and other things, and we just kept coming back to the mixed population as the ideal source. So the paradigm at the time was really to try to identify that one cell and grow it up in a petri dish. And I guess we sort of turned it on its head and said, if everything works, let's figure out what things are maybe harmful, the terrorist cells, remove those. Maybe we can extract something and make the population work better, but do we really have to find that one cell and grow it up? And I think over time we've just convinced ourselves that every time we've looked, the mixed population works better than a cultured population.
And Rhonda, maybe you'd like to comment about how many different flavors and formulations did Aastrom go through, and how do you QC at the end that your product is the right population of those that have been expanded and those that have been reduced?
Sure. One comment before I answer your question is, I think some of the argument, too, between a mixed population versus a purified population tends to go hand in hand with an allogeneic versus autologous, just because you put your product development hat. The complexity of trying to do a mixed population allogeneic, because the characterization of things you have to do for an allogeneic product are different than you need to for an autologous. So those two tend to go hand in hand to begin with.
No.
In terms of how did we come up on this particular combination of cells, it was basically where we got a maximum of the stromal cell component and the macrophage component. So if you look at a time course in terms of how the product develops over time, you start to see the M2-like cells come up at about 6-7 days and peak at about somewhere between 10 and 15 days. And you need that 10 to 12 days to see the MSC population. So we wanted to make sure we had, the majority of the product coming from those two cell types. So it was basically a time and culture scenario. In terms of QC, what we do at the end of the day is phenotypic analysis and by flow cytometry. We do, same thing everybody else does, count viability, sterility, safety, and those sort of things. And then we do just a number of assays for information only to help us narrow in on potency assays and those things going forward, which those I can't discuss too much, but it's mostly phenotypic and viability.
And Linda, when you made a decision to go from auto to allo, help me understand what happened when you started looking at cell types, because you're using a cardiac cell. And then I'd like you to expand a little bit for me on delivery methodology and the work you did, only because I know that Aastrom, I know that Mesoblast, and I believe Cytori as well are all using NOGA to deliver, and NOGA is kind of an arduous process. You're saying that maybe you don't need to use NOGA, so I'd like to understand that.
So in terms of the transition from autologous to allogeneic, we actually had well characterized. We have a mixed cell population as well, and we feel that that's one of the strengths of our preps. But we felt that we knew our cells well as an autologous cell. We continue to know those cells well as an allogeneic cell, and we're using the same standards for release criteria that we used with the autologous product. The difference, of course, is in our ability to do QA and QC. And in fact, that process failures don't affect a patient, and also the off-the-shelf ability of the cells. Plus, we get to know them even better because we can study them in 22 ways and know that this cell bank has these characteristics. And so we found that transition with a mixed cell population very easy. In terms of delivery method, it's actually an interesting story. We started in this business believing that intramyocardial delivery using the Noga-Myostar was the way to go. And then several years ago, CIRM, the California Institute of Regenerative Medicine, provided our academic colleagues at Cedars with a grant to compare intracoronary delivery of the CDCs to intramyocardial delivery of the Cardiosphere product, as well as the CDCs. And this was done in a large animal model using MRI, reduction of infarct size, and ejection fraction as endpoints. And what we found is that while intramyocardial delivery was comparable to intracoronary delivery, there really was no difference in terms of the efficacy of the cells using intracoronary infarct-related artery, is the way Jason likes to call it, IRA, or the intramyocardial delivery method using Noga-Myostar. And so what we decided is that it was actually easier and more adaptable and less time-consuming and less expensive to continue with the intracoronary method. And so that's why we've sort of made that shift. Now, in addition to that, recently published in Stem Cells has been a novel method of how the cells are taken up into the cardiac parenchyma using the vascular bed. So, if you have a chance, take a look at that paper because I think it explains why we think this is an important mechanism of action.
So to wrap up this panel, I'd like to ask our panelists if you have anything that I've left out or any just brief comments that you'd like to make in order to close. Why don't we start with Linda, and we'll just work our way back.
Actually, you caught me a little bit by surprise asking for final comments. I guess I want to say it's an honor to be on this panel. We were a little bit of a latecomer to this scene, so we've been able to take advantage of all the hard work done by our colleagues up here and derive a product that I think is the best combination of a good business model and a good clinical product at the same time. Silviu?
Yeah, look, I would agree. I think you've got to have a combination of commercial insight as well as strong science to get a product for cardiovascular disease in particular, because I think cost of goods are going to be critical when all's said and done. And we're talking about large numbers of patients. We have to have a product that's available easily, when needed, and in a cost-efficient manner.
Great. Marc?
I'd just say, while we tried to highlight some of the differences between us today, I think the more important thing is we're all really aligned at the hip in this industry. I think, to echo the words of the keynote speakers, there's a tremendous disruptive opportunity in regenerative medicine, and I think we're all aligned up here in trying to make this happen. And I think panels like this really help get the collective message out there that something exciting is happening up here in regenerative medicine. I appreciate it.
Thank you. Rhonda?
Yeah, I don't have too much to add other than, again, thank you to the Alliance as well as Jason for the invite. And to echo those sort of things, too, is I think it's the cardiovascular space for cell therapy has been around longer than a lot of them. It's really nice to see, a number of companies all in about the same space, or in that phase 2, phase 3 transition, that it comes back to the image of how are you going to help the patient, and how is this going to become a therapy and not just a science project.
Okay, so that's going to wrap up our panel. We're going to take a 10-minute break right now. 5 minutes, sorry. And I want to thank our panelists and thank the companies who will be presenting individually later today. Thank you.
Yeah, just 5 minutes, everybody.